So I'll be speaking at the ESH meeting about leukemias at the T-lymphoid myeloid interface. So this is a subject I've been interested in for a number of years, and we did quite a bit of work on this in my previous job, which was in France at the Hôpital Necker in Paris with Professors Elizabeth McIntyre and Vaid Esnafi. And in my talk, there's several sections. So first of all, I give people an overview of what we mean by immature TALL and how important it is to relate this to normal T-lymphoid differentiation, because that's really underpins mechanisms of leukemogenesis, and it's really a useful way of putting things in context and for asking questions about mechanisms of leukemogenesis. And then I go into a bit of detail on clinical and biological data for the early thymic precursor or ETP-ALL subgroup, so in children and also in adults. And again, this is the work we've done in Necker over the past couple of years, thinking about the mutational profiling of these adult ETP-ALLs, which surprisingly is very similar to children actually in many ways in terms of mutations in signaling pathways and hematopoiesis and epigenetics. Does have some important differences, particularly in the incidence of mutation in DNA methylation genes. And I talk about some important clinical considerations, for example, the use of stem cell transplant, the fact that the biology of ETP-ALL is quite heterogeneous. So, for example, HOXA overexpression probably confers a worse response to treatment. So in the second part of my talk, I'm going to present some new data, which again was carried out in Paris, supervised by Professor McIntyre and in collaboration with Elisa Laurenti at the University of Cambridge. So these are new data that has just been accepted for publication in Leukemia, so maybe online by the time people come to watch this or the ESH presentation, but I've also given links in my talk for seeing the full data. And this project was all about trying to use new bioinformatic techniques to think about leukemias a bit differently, and immature leukemias in particular. So we have this issue that in practice, we classify leukemias mainly by immunophenotype, which works very well for a lot of cases, but we all know that some leukemias can be very difficult to classify. So this includes immature leukemias, both immature TALLs and also immature AMLs, and that leukemias that are difficult to classify often do poorly. So again, this is the case for TALLs and AMLs. And really this classification system, it's very binary. So in practical terms, we give patients either treatment that's ALL directed or AML directed. But really, if we want to consider this in the context of normal hematopoiesis, there's this kind of part of that doesn't fully make sense, because we know that in normal hematopoiesis, it's not binary. So there's these kind of old notions of the lymphoid myeloid dichotomy, where there's an early branching into lymphoid and myeloid arms during differentiation, which we now know is no longer true. So we've known for a long time that there's retention of pluripotency even at fairly mature stages of development. So for example, on, let's say, the lymphoid half of hematopoiesis, even mature lymphoid cells can still retain myeloid differentiation potential. And this includes, for example, early thymic precursors. So we know that the kind of binary nature of this, or the dichotomy, is artificial. And we're interested in thinking about it in a way that's more coherent with our modern understandings of hematopoiesis. So, of course, that's been driven over the past 10 to 20 years by the genomic revolution. So, huge amounts of information about the genomics and transcriptomics, epigenetics, for example, of normal hematopoiesis. And in parallel with that, that's been aided by the development of new analytical techniques. So in parallel, there's been lots of bioinformatic advances and the use of new techniques to analyze these data. So really the starting point was wondering whether we could use some of these newer techniques to think about acute leukemia maybe slightly differently. So we undertook a project where we performed transcriptional profiling of a large cohort of acute leukemias. Just over a third were TALL, the rest were AML, and we very deliberately biased the cohort so that there'd be a lot of immature cases. And then in collaboration with Dr. Laurenti's group in Cambridge, we did a lot of bioinformatic analysis. So tried to use some of these newer techniques to interrogate the transcriptional profiles. So we did quite a lot of things, but in my talk, I focused specifically on a technique called ICGS, which is iterative clustering and guide gene selection. So this is an analytical technique which has been used mainly to analyze single cell transcriptomic data, and basically relies on the identification of a core repertoire of what's called guide genes that define coherent transcriptional modules, so kind of let's say the minimal number of genes that can define whole transcriptional programs. And the process involves serial iterations and serial clustering and identification of guide genes and refinement of this to arrive at the clustering pattern. And I think the thing, as I say in my talk, the thing that I find kind of appealing about it is that if you look at data using this technique, so for example, in the original paper that described this in Nature a couple of years ago, it gives what I think is a more realistic vision of hematopoiesis, in that in contrast to some clustering heat maps that you see that are very neat boxes, that everything's quite tidy. I think it shows nicely the kind of dirty or blurry transition as cells progress through hematopoietic differentiation, and the fact that within hematopoietic precursors that there's actually very heterogeneous activation of various transcriptional programs. So we thought this would be a good technique to apply to our cohort, which we did, and I showed these results in the presentation. So there's quite a lot of info on the heat map, so anyone watching it, I think it's worth pausing on some of the slides and just worth digesting the images, because there's a lot there. Essentially what we found that was different to other clustering techniques is that we showed a spectrum of differentiation. So on the one hand, we had a T-ALL cluster that had high expression of T cell genes, and then on the other extreme we had a couple of myeloid clusters with high expression of myeloid genes and no expression of T cell genes. But then in between, we saw these kind of intermediate clusters, which we ended up calling interface clusters, in these cases interface acute leukemias, that had either very low expression of any guide genes or were partially myeloid. So we were interested in pursuing these clusters a bit further to seeing what they were about. So we saw that when we used other bioinformatic techniques to classify immaturity, that there was an overlap with this. So by other methods such as dimensionality reduction with PCA, we found that there was an incomplete overlap with, for example, ETP-ALL status, and also that the mutational status of these cases partially overlapped with what's normally observed in immature leukemias, but not exactly. So we went into some detail looking at the transcriptional profile of these cases, mainly by gene set enrichment analysis, and we wanted to work out what normal populations these leukemias resembled. So we performed GSEA with a series of previously defined hematopoietic progenitor populations, and also some new data which we generated during the course of the study, which was thymic transcriptomics at different stages of normal thymic differentiation. And what we found was that these interface cases, so these IALs, which I'll call them from now on, had transcriptional programs that resembled both myeloid and lymphoid competent progenitors, which we kind of anticipated because it's this mixed group that's in between myeloid and lymphoid. The thing that was surprising for us is that when we looked in more detail and worked out what was driving the resemblance to the various individual hematopoietic progenitor populations in each of these clusters, we found that it was cases originally diagnosed as AML that drove the resemblance to lymphoid populations. So to put that another way, it seemed that within these IALs, cases that were AML, in inverted commas, were actually more lymphoid similar, let's say, than the T-ALL cases. So this was a surprise. And this was also true for some analysis we did in mixed phenotype acute leukemias, that it was really the myeloid cases that were more lymphoid than the lymphoid, if you see what I mean. So we then went on to see whether these type of signatures might have some prognostic significance. So we did find this in AML cohorts. So we calculated something that we called an IAL score, which I won't go into detail on, but essentially it's based around transcriptional resemblance to the interface acute myeloid leukemias in our cohort. So basically the genes that differentiated them from the other AMLs in our larger cohort. And we calculated this score for 2 independent cohorts. And what we found is that AMLs that had a high IAL score, i.e. cases that were transcriptionally like our interface cases, or IALs, had a very poor outcome. So this is in 2 independent cohorts that we analysed. They had greatly reduced survival. And this prognostic effect, we had extra data for one of the cohorts, the alpha cohort, which was carried out in France. And we were able to do some multivariate analysis in collaboration with our clinical colleagues. And we found that this IAL score was strongly predictive of prognosis and was independent of other strong prognostic parameters, such as cytogenetics, for example. So this was interesting from the prognostic point of view. And then the other kind of intriguing part of that was that when we looked at the high IAL cases in both those cohorts, we found that they also had transcriptional similarity to immature lymphoid cells. And in particular, they each had expression of a B lymphoid signature. So this is kind of intriguing. And again, in the context of the pluripotency that's involved in normal hematopoiesis, we wondered if this indicates that these cells actually retain a lot of lymphoid identity, or potentially maybe even in many cases arise from multipotent or pluripotent B myeloid precursor. And it begs the clinical question. I mean, I think it's a very kind of preliminary idea at this stage. But if these cases are not doing so well with AML-directed treatment, whether at some point ALL-type treatment might help or contribute or could be implemented in some way. And I do highlight in the talk that there's a very interesting paper from the Sauvageau group in Quebec from a couple of years ago, where they showed that AMLs with RUNX1 mutations, so typically immature AMLs, as you know, are responsive to corticosteroids. So this is intriguing. I mean, we found this in our cohort as well, that there was a lot of RUNX1 mutations in the interface cases. So whether there may be at least a theoretical argument for thinking about adding this type of treatment in these cases. As I say, this is a suggestion, needs to be tested maybe in the laboratory first, of course, but it's kind of a bit of a tantalizing prospect. So they're the main topics I discuss in the talk, and really the overarching messages are that, number one, that thinking about these leukemias, it really needs to be directly linked to normal differentiation processes, and that in thinking about normal differentiation processes and modern understanding of hematopoiesis, that we can harness these new ideas and these new techniques to maybe think a bit differently about some cases that are difficult to manage in the clinic.